I guess I will start. Can you hear me? Good. So what I'm going to tell you about is a part of the work that we're doing in the lab, which is following up from some of my postdoctoral work, which I did with Chris Marks. And the subsequent work that has happened in the last year or so since I started working here at NCBS was done largely by Mridula and Kritika, were two graduate students in the lab. And I think most of you have probably seen or met Mridula already in this meeting here. So I'm going to start off by just saying, just for Brian, that synonymous mutations are not neutral, and there's like a not always neutral, really, is what I want to say. They just have very variable effects, and that's partly what I'm hoping to show you today. So why do we care about synonymous mutations? Again, most of you have already heard about this already. We know that there is strongly biased codon usage across species, and ever since one of the first DNA sequences started coming out, even bits of them, people were noticing that there's variation in which bases were used more or less frequently, which codons were used more or less frequently. And people hypothesized that this was because there was a role in translation or transcription that these codons were playing. And over the years, there were a lot of very nice experiments done showing that if you switch some codons in E. coli, for instance, in specific proteins, you can modify the solubility of that particular protein. We know also from many heterologous expression studies that such synonymous mutations, either alone or in conjunction with other non-synonymous mutations, can have large effects on solubility of proteins, their stability, how they're transported, sometimes how they're spliced in the case of eukaryotes. And yet we sort of don't really have a very good general understanding of what is it that causes biased codon usage across species. So what are some of the predictions that we have from theory? And my references seem to have disappeared, but I'm happy to tell those to you later on if you'd like. So some of the earliest work suggested that there might be weak selection acting on most genes and stronger selection on just a few genes in terms of which codons are used. So the idea would be that, for instance, people notice that there's a very strong correlation between gene expression level and codon use. So highly expressed genes tend to be more strongly biased. And so the idea would be if these are genes that are under strong selection, they're sort of dragging along the rest of the genome, which by itself is rather weakly selected in terms of its codon use. There's another bunch of work that suggests that maybe it's stabilizing selection that's acting on codon bias instead, and there's been some recent work of comparative sequence analysis that suggests that as well. And then what it ultimately boils down to is some of these old models from Bulmer and Paul Higgs and other people have shown that there might be coevolution with tRNA gene copy number. So the basic idea is, so you find this strong correlation between tRNA gene copy number, which also correlates with the actual number of tRNA genes present at any point in the genome, in a cell. And the idea is that if you're using codons that are more frequently, whose tRNA counterparts are more frequently present in the cell, then it's more advantageous because you don't have to wait around so long to find the right amino acid and incorporate it. So here's a summary from a review paper from a few years ago, and about what are the possible physiological or cellular mechanisms of these effects, right? So I'm sort of transitioning here from some basic population genetic understanding of what might be leading to these evolution of codon biases to trying to understand what the physiological implications of these codon biases might be, because I think they inform each other, and we need to know what the mechanisms are to really build theory that's rooted in what might be happening physiologically in cells. So one possibility is that mRNA stability, I'm sorry you can't quite read this, but I'll walk you through it. So mRNA stability might be affected by which codons are used in a particular transcript, and that stability may determine how that mRNA is then, how stable it is, and how it gets expressed eventually or translated. There's also one hypothesis that suggests that codons at the beginning of the transcript, it's important to have rare codons, so that, and you don't want to have very frequent codons, so that there's this ramp up of translation speed, and translation initiation is not a problem, and there's no more traffic jams towards the end. Another idea suggests that if you have ribosomal pause sites in the middle of the transcript, so if you have, for instance, motifs that will be like Scheindal-Gardner sequences, which is where the ribosome binds at the beginning of translation, if you have these in the middle of the transcript, then you're going to inhibit regular ribosome movement during translation. And that may be what is problematic in terms of deciding what codons should be used or not. And then finally, with bacteria, if there's co-translational folding, so as a protein is being translated, if it needs to be folded up correctly, it may sometimes actually, you may need some pause sites in that transcript so that everything slows down, the right fold can happen, and then the protein can sort of go along and move on its way. And we know that, for instance, from some comparative work, people have shown that in large proteins where there are big domains that need to fold over, at those domain boundaries, there's often rare codons, suggesting that sort of slowing down of the ribosome is important in that case, and so you would want rare codons to be used, right? So there's a large number of possible mechanisms, and what's likely true is that there isn't a single mechanism that operates across all different species that we understand. And I'm going to take a very bacterial sort of microbial prokaryotic approach here and not talk too much about eukaryotes. You've heard already about some eukaryotic work from Brian in his lectures earlier. I think Hiroshi is also going to talk about some work. So what are the major questions that I'm asking? One is, what is the nature of selection acting on synonymous changes in functional genes? So if we can measure experimentally what is the strength of selection, under what situations or scenarios do we actually get strong selection acting on codons, maybe we can then use different methods to infer what is the nature of selection acting on functional genes in terms of codons. What mechanisms underlie these effects? And like I said, I'm guessing there's a diversity of effects. We want to know what they are exactly. What is the evolutionary response to selection? So if we do find that there is selection on codons, how can we understand how are species going to respond to it? And finally, how do these effects shape genome evolution? So if we agree, as data accumulates, that codons are indeed cumulatively a large sort of selective driving force, how does that shape genomes in general? And can we come up with some overall conclusions that might help us understand species and their genomes overall? So to test this, I started this work as a postdoc, as I mentioned earlier, with Methylobacterium extorquens, which is a benign species with a giant genome, lots of crazy stuff otherwise in its chromosomes. But I'm focusing here in today's talk on one gene. This is formaldehyde activating enzyme. It's located, so this is the main chromosome of Methylobacterium. It's sort of, not too close to the origin and not too later on as well, which is also has interesting effects on gene expression and evolution. And so I've highlighted FAE here as part of its operon. Well, it's not really part of an operon. That's what I want to highlight here. So whatever I do with FAE is unlikely to be a result from what happens in nearby genes. And so what FAE does is it helps this organism essentially use single carbon compounds as carbon substrates. So it's methanol as an example that I'm showing you here. Anyone that uses methanol has to sort of essentially go through this process of converting into formaldehyde, which we know is toxic. So FAE binds to this formaldehyde and detoxifies it, converts it into something that's not so toxic anymore, right? So without FAE, these cells cannot survive in and use single carbon compounds as carbon substrates. And what's nice is that we know that Methylobacterium is one of these relatively few species of bacteria that actually use single carbon substrates in nature, that have evolved to do that, to be able to do that. So we know that this gene is under selection, should be under selection in its natural habitat. So I created a few different synonymous variants. I don't want you to focus too much on the details. I'll just describe the main point of this quickly. So what you see here is the N-terminal part of the gene. Here's ATG, which stays the same for all of my alleles. Here's wild type, and what I've highlighted is the rare codons. So what I'm calling rare codons are codons that are underrepresented in conserved sites or conserved amino acid residues across the genome, across the protein coding sequence of Methylobacterium. So orange are rare codons that are naturally present in the wild type allele. Blue are frequently used codons, and frequent again is across the genome. Gray are intermediate codons, so when you have more than 2 synonymous codons that will give you the same amino acid, there's a third possibility, right? You could get the most, the best and the worst, like frequent and rare, but you also have these intermediates. So that's what these grays are. And what I've shown here then is here's one allele that I recoded essentially to make completely frequent, right? So it only has frequent alleles, frequent codons. Similarly, there's a variant here at the other extreme, which has only rare codons now, right? And this sort of continues through the rest of the gene. And then there's various intermediates in between, which have 50% rare and 50% frequent in different combinations and placements. And so this is just to see what kinds of effects would we see. And when I started this experiment, I had no idea how much of a big effect I would find. Would I find anything after all this cloning and doing all this stuff? If I don't get any result at all, then I was like, my only postdoc. So this was sort of, going all out and hoping that would find some effects. And as it turns out, the effects are huge. And now I wish I had some single mutants that I could, I could have worked with. But anyway, so what's happening essentially is the mutants are making less FAE enzyme than the wild type. So what you see here is protein per mRNA. I think I'm missing a slide, but anyway. So here's the wild type, and here are all the different mutants you see in different colors. And what that results in is that growth rates of the mutants is much lower. So the rest of this talk, I'm going to focus on growth rate as a fitness proxy. So what's interesting then is that this is the allele which has only frequent codons, and that's the one which is almost as bad as the knockout for FAE, which is shown in the dashed line here, right? And here's another allele which is also very similar to this one, except the active residues of this protein are now changed to be rare codons. So what we see is in situations where this sequence and this sequence are very, very similar to the wild type sequence, which is 70% frequent anyway, and that they seem to have been the worst hit by these codon changes that I made. So that was odd and strange. What is also interesting is that the fitness effect is substrate specific. So you don't find a big fitness effect in succinate, which is a multi-carbon substrate. So all of the different strains have the same fitness in succinate, but the fitness in methanol varies, like I showed you earlier. So that previous data was for a single carbon substrate. That's the only place where there's an effect. And what this suggested is helps us rule out a few other hypotheses, right? Because if it's a genetic problem with, for instance, sequestering ribosomes, which is one of the hypotheses that have been predicted, well, then this couldn't happen. Because in succinate also, FAE is still very highly produced. So in methanol, FAE is expressed about 2% of cell protein, which is very big. And in succinate, it's still about 1% of the cell protein, right? So it's kind of not very well regulated in general as genes go. But in succinate, if 1% of your cell protein is kind of messed up, it's bad, it's misfolded, it's sequestering ribosomes, that should reflect in fitness, and it doesn't. So suggesting that sequestration may not be a big problem. And then I want to tell you about studies that have come out since I did this work, which also tell us more about what are the more general effects of different codons. So here's a recent paper where both are from George Church's group at Harvard. What they did is they changed, in this particular case, the first 11 N-terminal codons, right, and made them either rare or variant in E. coli, and this is with GFP. And they saw that the fold change in protein expression is much lower for common codons than for rare codons, right? So rare codons at the beginning are better in terms of higher gene expression. So that's one thing. The other study that also was published from the same group with a few different people is that they recoded all of the rare codons in about 42 genes in E. coli again. And these are all genes that E. coli cares about. And what they find essentially is if they replace each of 13 forbidden rare codons throughout these 42 genes and then look at the doubling times of the resulting E. coli strains, what they find is the recoded ones have a slight defect compared to the kanamycin-resistant sort of cloning control that they use, right? So compared to the control, the recoded strains on average have a slightly higher doubling time. Okay? And this is another the third point is just showing you genes which were recoded using a different method. So there's just a methodological difference in these two. So again, what all of these are suggesting is, what I'm finding hopefully is just not, in my case, with Metallobacterium, with this one gene, it's likely to be more frequently found that you will find effects of recoding specific codons. So what about distribution of fitness effects of synonymous mutations? I think at this point everyone here knows what DFEs are, right? So here's another study actually published about the time I was finishing off my work, was that which shows that the distribution of fitness effects, so what they did was they just made random mutagenesis in 2 different ribosomal protein sequences in Salmonella. And what are shown here are the frequency distributions of the fitness of these single synonymous or non-synonymous mutants, right, for these 2 genes. And so what you see is the relative growth rate in LB, which is a rich medium, and a poor growth medium. And this is for synonymous mutations. This is for non-synonymous mutations, right? So they're very, very similar. So you can see that the tail is a little bit fatter for synonymous mutations, a little bit different for non-synonymous, and so on. But, I mean, overall, there's really not a huge difference, suggesting that at least in the case of very, very important central genes like ribosomal proteins in Salmonella, it would be very, very important. Synonymous mutations could be as important as non-synonymous mutations, almost to the same extent. Okay, so another piece of data that I had from my work was that if I now put all my different alleles in a plasmid and overexpress them, would that rescue the problem, right? So we should again help to get a mechanism. And so indeed what you see here is, this is on the x-axis is the concentration of inducer, which is just tuning the expression of this gene up and down. As you go to the right, you're increasing expression levels, and on the left again is growth rate. What you see is that most alleles show this increasing trend in growth rate, so they can be at least partially rescued by just making a ton of this already highly expressed gene, right? Except for this strain, which doesn't respond at all. So that's another story. So what this suggests immediately then is one way to fix this problem of not having enough enzyme is to just overexpress this gene, right? So to see what happens then, I did a laboratory selection experiment, took Metallobacterium, which is unhappy to start with, evolve it in batch culture, and hopefully you get a happy bug at the end, which we did get. And so what I'm showing you here is data for growth rate and change in growth rate across a number of generations. So the generation is about 150 or so for some of them, and then some of them evolve much faster in about 60-odd generations, right? So each panel is a different mutant that was evolved. Each solid or dashed line is the replicate population. And the dashed lines are repetitions of these initial experiments that I had done in Boston, right? So the black lines are what I did in Boston, and the dashed line are what my students did last year here in India. So it's kind of nice to see that they're kind of parallel, and it wasn't a freak accident of what I was finding earlier. So what you see is that everyone rapidly evolves to have higher fitness, in some cases as good or better as the wild type, which is shown in the dashed line. And I also evolved wild type in the lab just to be sure that any adaptation I'm seeing is not sort of media specific or protocol specific. So then what happens in all of these, right? What are the mutations that are responsible for it? What is the solution? What I find is a lot of mutations or synonymous or non-synonymous SNPs segregating in all of these different populations. So here's one mutation, which is synonymous, by the way. This is the line that had 100% frequent codons, and it only gets hit again and again 9 times now in 9 different replicate populations with this one single synonymous SNP, which, as I'll show you in a minute, has a huge effect on fitness. So you go from, selection coefficients are really, really large. This is another strain where 2 different mutations were picked in separate lines. And so all these mutations I'm showing you are never occurring together, by the way. So each line only has one of these mutations, one of these X's, right? So there's another line here which has a mutation just before the start site in the putative ribosome binding site, perhaps. So maybe what's happening here is perhaps these mutations here are upregulating this gene, right? So I'll show you some data on that. So are these mutations beneficial then? So what Mridula and Kritika have done is taken these single synonymous or non-synonymous SNPs and moved them back in the ancestral background, or moved the ancestral allele back into the evolved background to figure out what's the relative contribution of all of these mutations to fitness. And so what you're seeing here is growth rate. Here's the wild type growth rate. Here's the ancestor. Remember, this was the one that doesn't really grow very well at all. So I'm only going to show you data for one strain just to simplify things. Different strains do slightly different things. You see the evolved isolate. So this is a single clone that is isolated from one of the evolved lines at the end. So we played it out and just pick one colony. So you see that the evolved isolate is quite fit. Here's the effect of the single SNP alone in the ancestral genome's background, which is indistinguishable from the evolved SNP's fitness. And then finally, the evolved background in this particular case also has a large effect on fitness. I think it is significantly smaller than that of that SNP, but by not by too much, right? So now if there have been other mutations in the genomic background, we know that from some genome resequencing that we've done. But the question is, are these single SNPs sort of more important for adaptation, or are there other mutations in the background that are more important? What you can see is if you expected that these fitnesses were additive in both cases, you'd get a much higher fitness for the evolved isolate, which you don't, right? So they're not acting synergistically. They're antagonistic in some way, and we don't know yet in what way. And so this picture sort of changes a little bit based on different SNPs that you look at. If you move one SNP for each of the evolved SNPs that we find, in some cases we find that the background has no effect at all. And that is what actually we see in the one case that I pointed out earlier, where it's just the very, very frequent allele, right? The frequent codon allele that just whoop, goes to fixation like that, increases in fitness really rapidly. There's only a single SNP that's responsible for all of the fitness effect that we see, right? So the picture changes based on what alleles you're looking at. And then we also look for, well, are these mutations then affecting protein production or mRNA production? And you see that the results are quite variable. Again, so here, focus on the mRNA data first. Here's the ancestral mRNA production, pretty pathetic compared to the wild type. The evolved strain has a little bit higher expression of the mRNA. The SNP alone actually gives you much higher levels of mRNA expression compared to the evolved isolate. And then the background also is pretty high. But the evolved isolate is producing far less mRNA. And what I don't know here, of course, is whether the mRNA is being produced and just getting degraded really fast, or it's getting translated, and I don't see it in the qPCR assays. I don't know all of that quite yet. Also for the protein, interestingly, we find that the SNP does not have a very big effect on the amount of protein, but the genome background mutations do have a large effect on amount of protein produced. But so the SNP basically has different effects on mRNA and protein, so that if you look at the ratio of protein per mRNA, it varies quite a bit for the evolved isolate, for the SNP, and for the effect of the background, right? So what we're getting from this then is that the effect of mutations and backgrounds, all of them can compensate for the initial fitness defect, which was caused by synonymous variants. So to summarize then, synonymous mutations likely have a wide distribution of fitness effects, which are also dependent on the substrate, on the environment, and also on the specific local context of the DNA that they're residing in, or the mRNA that they're part of. We see parallel yet allele-specific evolutionary solutions. So what we're doing is also moving. So some of the alleles, the ancestral alleles, had very similar sequences early on in the 5' end, right? And all of them could have hit the same mutation because it was possible to get that same single SNP. But only one of them, one of that ancestral mutant, consistently gets that SNP. All the others do not. So why is that the case? So is it that there are some other mutations in that local context of that allele that make it so that this one is not beneficial? So to test that, we're moving SNPs around between ancestral alleles as well. And we see that a common phenotypic problem, which is the lack of functional enzyme, can be solved in very different genetic ways, sometimes by potentially upregulating it through some regulatory or promoter level mutations, sometimes by changing the transcript perhaps, or translation level machinery, or how it's functioning. And so short-term coevolution between tRNA genes and codon users seems unlikely, right? So through genome resequencing and so on, we haven't really found any tRNA genes that have been mutated in any obvious way, or they're promoter mutations or something. No gene duplications or deletions. And so it seems that if an organism accumulates a few synonymous mutations over time, it's unlikely that in the short term, the response is going to involve any change in tRNA, right? So it's still a bit of a mystery why we see this tight correlation between tRNA gene copy number and codon usage. If when we change codon usage, we don't see a tRNA gene copy number response. And the opposite question has not been addressed yet. So recently, a couple of papers came out where they manipulated the number of tRNA genes, so knocked out a number of tRNA genes, and sort of looked at the fitness effect. Now, they need to like evolve those lines and then see what happens to codon usage. Does it eventually change? Problem, of course, is codon usage can only change in small increments at a time. And to get a big effect on codon usage, you'll need a lot more time. So yeah, I think I'm going to stop here for now. Question? Not within this coding sequence, no. No, not in the FA region. So we do have, I think, a few synonymous mutations that show up in the rest of the genome, but we haven't tested all of their fitness effects yet. So it's, yeah. You mean so for this particular species, or even like species within the genus? Yeah, yeah, we can do that actually. I think there are some. What I can say though is for this gene particularly, we have about eight methylobacterium species fully sequenced, full genome sequences, and if I look across them, this gene is very highly conserved. So it's been thought that this gene has moved through horizontal gene transfer at some sort of older time period, and it's been retained because they use it quite a bit. It's very important. So it's possible that codon bias in other species that are related, closely related, very closely related, is a little bit different. Actually, I think it is different at the genome scale because the genomes themselves vary a lot. So this one has a giant genome, with all kinds of plasmids and lots of IS elements. Very close related species that Chris also works on now is PA1, which is just very small genome, nice and streamlined, and so on. So, the number of tRNA genes also varies quite a bit across these strains. But FAE seems to be very highly conserved. Yeah, some don't. Yeah. In this case, yes. In the situation that I'm looking at. Yes. Yeah. 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 That's true. So actually, this is based more on sort of the other studies as well that people have done, like looking at DFEs, right? So there are some synonymous mutations don't have any effects, and they are single mutation studies. Some do so. For the now.